This is a G protein link receptor, which is called that because we have a G protein that's inactive. So when the signal goes in, the G protein diffuses across the membrane to the receptor, and the GDP gets exchanged for a GTP, and then it becomes active. And when the signal is done, the signal will leave, and then the G protein will diffuse its way across to the enzyme, which then activates the enzyme. And since we don't need that forever, it changes back into GDP and we have a phosphate. And it's here we have receptor tyrosine kinase. It's an amino acid that makes two polypeptide chains. As the signal comes in and binds to the signal binding site, the two chains come together and form an unphosphorylated dimer. And then we get ATP, and it takes the phosphorus from that ATP and attaches it to the tyrosine, which other proteins come and get phosphorylated, which activates the proteins and creates a cellular response. And we can have multiple on the same dimer. So here we have a ligand-gated ion channel. So there are ions on outside of the cell membrane, and we have a signal, but the channel is closed. So the signal will attach to the binding site of the channel, which, as we can see, it opens up the channel and allows all the ions to pass through safely. But there will be a time where the signal will stop because it won't last forever. So when it leaves, the channel closes again and not all the ions will be able to go through. And that's a cellular response.